Okay guys, so today we are going to go over the transport of materials into and out of the cell through our cell membrane. Talk about the cell membrane a little bit more, talk about all the different structures in it, and then we'll go over all the mechanisms. So to start off, we're going to go over diffusion. And diffusion is very simply going to be the movement of molecules into and out of the cell from an area of high concentration to an area of low concentration. Now, when we talk about simple diffusion, which we're going to start with, we're going to talk about very easy things. We're going to talk about nonpolar things going right through our cell membrane. So here's the example your book gave. I'm not going to go over this example too much. I'm going to do one on my own just because I think it'll be more useful for us. But if you notice, in this diagram here, we have water traveling through some membrane. Now, it is not the cell membrane in the book's example, but the goal of this diagram is just to show you that water is moving from an area of high concentration on the left side here to an area of low concentration on the right side. Now, we can also have more than one solute particle going each way as well. So you can see there's yellow ones on this side, there's purple ones on this side, and they are going to keep moving past whatever membrane this is until we reach something called equilibrium. Now, equilibrium is when we have an equal concentration of our solute to our solvent on both sides of our membrane. And again, let me just repeat that. It is equal concentrations, not equal particles. It's equal concentration of our solute to solvent on both sides. So let me draw picture for you of our cell membrane, because I think this will be more useful for us than the book, its diagram. So I'm going to start off with the cell. There's our cell. Cool. What I'm going to do is I'm going to put our cell in some kind of beaker. I know my diagrams are, wow, that is just tragic. Let me redo that. Okay, so let me make a beaker here. That looks a little better. There we go. Okay, so there's my beaker. Here's my cell. We'll throw it in some water. Why not? So what I'm going to do is I'm going to make some solute particles. Okay. Now, I'm making a bunch on the outside, and I put a few solute particles inside the cell. So, that is a solute particle. Okay. What is the solute particle? Well, in this example, let's go over something small, nonpolar, like oxygen. We could do oxygen. It is small and nonpolar. Okay. Now, if we have something that is small and nonpolar, it can pass right through our phospholipids. Passes through our phospholipids. Okay. And again, that's if we have something that's small and nonpolar. If it's something that is large or something that is polar, yeah, it's probably not going to be able to pass through our phospholipids, but in this case, it can. Okay. So here's what's going to happen. We are going to have a lot of solute particles on the outside. We're going to have a little bit in. So our solute particles are going to go into the cell, passing our phospholipid bilayer, right in between our phospholipids. Okay, they're going to go in. Okay, so solute goes into cell. Until equilibrium is reached. Does that necessarily mean we are going to have the same amount of solute particles inside compared to outside? No, it does not. It's a concentration of solute to solvent. So if you look at the area that the cell is in, we are going to have a lot of solvent out here. We're going to have a lot of water. We're having a smaller concentration of water inside the cell. So we just want that solvent to solute ratio to be the same outside and inside. So we might not have the same amount of solute particles inside or outside, guys, but we want the concentrations to be the same. That is diffusion. That is it moving from an area of high concentration to an area of low concentration. So in order for diffusion to happen, guys, we have to have a concentration gradient, okay? What this means, concentration gradient, is we are going to have a different concentration on one side compared to the other side, okay? So on one side we're going to have a higher, on one side we're going to have a lower, and that's how diffusion works, okay? In passive transport, so here I'll label them for you here. In passive transport, this is going to be simple diffusion, it's going to be facilitated diffusion. This is a word we'll go over shortly. Facilitated diffusion. And lastly, osmosis. Okay. In all of these, we're moving from an area of high concentration to an area of low concentration. Given that, we do not need energy. We do not need ATP for these three types of transport to occur. So passive transport is made out of these three types: simple diffusion, facilitated diffusion, and osmosis. So during simple diffusion, guys, we're going to be moving solute particles. That's simple diffusion. We are moving the solute particles from an area of high concentration to an area of low concentration. And that is when they are able to pass through our cell membrane. Somehow they're getting in or getting out, whatever way they have to go in order for equilibrium to be reached. Now, osmosis is a little bit different. Osmosis is going to deal with the movement of water. This is going to be moving the solvent instead of the solute. So in simple diffusion, we're moving solute. Osmosis, this is the only one where we are moving solvent. 99% of the time, we're moving water, okay? So here's the example the book gives you. We have a container here, okay? We have a selectively permeable membrane in the middle. That selectively permeable membrane will allow the solvent to pass through, but not our solute. Okay. For some reason, solute particles much too big or too charged, too polar to pass through. So we're going to try to even out the concentration of our solute to our solvent. So what's going to happen here? What's going to happen is our water is going to move, so our solvent, which is water, moves to where there is more solute. Okay? Now, why does it do this? Well, again, we're trying to even out the concentrations. So wherever there's more solute, it has a higher concentration of solute to solvent. If we move the solvent over, it's going to weaken or dilute that concentration. Okay, think of it like Kool-Aid, guys. Okay, if you're making Kool-Aid, you put some powder in, you put some water in, you put some sugar in. Let's not worry about the sugar right now. But if you put a little bit of powder in, and you put a little bit of water in, and you taste it, and it's like, oh my goodness, that is so tart. You know, you might put a little bit more water in. You're diluting the Kool-Aid, okay? You're diluting the Kool-Aid. That's how osmosis works. Same thing. Okay, so we're going to go over three different terms for osmosis here: isotonic, hypertonic, and hypotonic. These ones talks about solutions. We're going to do the solutions and the cell, just in case if on the test it gives you one or the other. Okay. Tonicity is the ability of the surrounding solution to cause a cell to gain or lose water. We'll go over a little bit of that later. But here is different solutions. So the first one over here, this is when a cell, in this example, a red blood cell, is put in a hypotonic solution, and we'll talk about what that is shortly. But when it is put in a hypotonic solution, that cell is going to get bigger and bigger and bigger. And if the concentration originally is too far off from outside to inside, that cell will get so big where it will break, it'll burst. Okay? The second one is isotonic, where the concentration outside the cell and inside the cell is roughly the same. Okay? So water goes into the cell and water moves out at the same exact rate. Nothing happens. Cell stays the same size. The last one is going to be a hypertonic solution. This is going to cause the cell to shrink, to shrivel. Okay? And it could get so small that the cell actually collapses on itself. So let's go over these three terms. Let's use them a little bit. I will start off with using solutions. So we'll say the first one is a hypotonic solution. Now for osmosis, what's going to happen is the solute particles are much too big to pass through the cell membrane. We'll do hypertonic. So, secondly, so instead of the solute particles being able to pass through the cell membrane, we are going to pass water through. Okay. And then the last one is isotonic solution. Okay. Okay, let's fill these with water. There's my water line, water line, water line. Drop some cells in here. Cell, cell, and cell. Okay? So we have two different parts of our solutions here. We have the solution surrounding the cell. That's the solution that we're talking about in the prompt for the question. So the hypotonic solution, that's referring to the area, this area right here, this area outside of the cell. Okay? If we said a hypotonic cell, that's referring to the area inside the cell. Okay, so two different parts. We have the solution outside the cell, and we have the cell, which is the cell. Okay? So let's put some solute particles in. Okay? So what's going to happen here is we're going to have a hypotonic solution. So let me put some solute particles in. Okay? Now, hypo means below. Hyper means above. Okay? So if we have a hypotonic solution, that means our solute particles are below or less than the amount inside the cell. Okay? So let's just dive a little bit deeper into this. In this example, if we have a hypotonic solution, that means that the cell is going to be the opposite of that. So the hypotonic solution means that we have a hypertonic cell. Same thing with this next one here. If we have a hypertonic solution, that means we have a hypotonic cell. So they're flip-flop. They're opposite of one another. Okay. Now what's going to happen is our solvent, this is osmosis, our solvent is going to go wherever there is more solute. So in this one here, the water is going to go where there is more solute. Well, is there more solute in the solution or the cell? You can see by the picture, there is more in the cell. So our water is going to go in, and the cell swells, gets bigger because of the water going in. Okay? And I didn't need to give you a picture for you to be able to solve this question. I could say I put a cell in a hypotonic solution, and I could say, what's happening to the cell? You would have to draw a picture. I would draw a picture. I think that's the easiest method to do here. So if you draw the picture, you have a hypotonic solution. That means the cell is hypertonic. That means the cell has more solute. You would just draw a picture, more solute in the cell, you're done. Okay? The second one here, we are going to have a hypertonic solution. That means a lot of particles are in the solution. Good. And we're going to have very little inside of our cell. Well, what's going to happen here? The water is going to go wherever there is more solute. Don't forget, guys, our bodies are made up of what? 70% water? So given that, we have water inside of our cells. So the cell has water all in it, all in that cytoplasm area. So the water is actually going to leave the cell and go out into the solution to try to get that concentration of solute to solvent to even out. In this one, the cell is going to shrink. And the last one, isotonic, you could say it's an isotonic cell too. Isotonic solution, isotonic cell, this is like the same. That means we are going to have the same concentration of solute to solvent inside the cell as outside. Now, I get it. Look, there is more solute particles. I drew 1, 2, 3, 4, 5, 6, 7, 8 outside, and I drew 1, 2, 3, 4, 5 inside. I could even do maybe a few more out here, okay? This is still isotonic. It's the concentration is the same. We have the same amount of solute to solvent, okay? So, water enters and leaves cell at the same rate. Overall, cell stays the same size. Okay? So there is your hypotonic and hypotonic solutions. Okay? So again, guys, if they could ask you either one. They could say, hey, the cell is hypertonic instead of saying the solution. If the cell is hypertonic, then you know the solution is hypotonic. It's always going to be the opposite, okay? All right. So this one, Osmo Regulation, just a definition, guys, the control of solute concentrations and water balance, okay? That's regulating our water levels. The paramecium, it is hypertonic, so this is like a cell that is hypertonic to its environment, okay? So here's your paramecium. We said that that paramecium is hypertonic. So I'm just going to write hyper in here. That means its outside solution is hypo. So we have a hypotonic solution and a hypertonic cell. If we were to draw it, I'll use a different color here, we'll use red. That means there's going to be a lot of solute inside of our cell. And a paramecium is a single-celled organism. And there's going to be very little solute outside. Yeah, we could even draw some more in here. Get a little crazy with it. There we go. So, which way water is going to go? Well, it's going to go where there's more solute. So the water is going to go into the cell. Okay? Water goes into the cell. If we have water going into the cell, well, our paramecium is going to get bigger. This is an issue because we don't want our paramecium to get too big. If the paramecium gets gigantic, it's going to explode. Okay? The organism's then dead because it's a single-celled organism. It's not like us. If one cell dies, we just, let's just make some new ones. Okay? Our paramecium cannot do this. It's dead after that. So what it has this thing here. This is called a contractile vacuole. And what that contractile vacuole is going to do is as this water is rushing into the cell because the cell is hypertonic to the outside solution, which is hypotonic, that contractile vacuole is going to get bigger and bigger and bigger, and it's going to fill with water. And then once it fills with water completely, it's going to squirt. Okay? And when it squirts, if you look here, we have these little things here. Okay? These little valleys or passageways. Okay? What that's going to do is that's going to literally push, using energy, using ATP, it's going to push that water outside of the cell. Okay? Now immediately when it pushes it out, it had then deflated the contractile vacuole, and right after that, well, we're still in a hypotonic solution and our cell is still hypertonic. So that contractile vacuole, once again, gets big, pushes the water out. Gets big, pushes the water out. This happens many, 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 many times a minute. If the paramecium does not do this every single minute of its life, it will die. It will get big. It'll fill up with water. It will explode. Okay? So that's why the contractile vacuole is a very good evolutionary adaptation for our paramecium. Okay, some vocab words here. If we put a plant cell in a hypotonic solution, meaning the cell is hypertonic, that means that cell will get bigger, all right? And it'll get so big until the cell actually resists taking any more water in. So the cell is nice and plump if it's in a hypotonic solution. We call that cell, it is turgid, okay? So it's filled with water. If it's isotonic, it's the same. That plant cell is not going to fill up with water. And that plant cell will become limp, okay, or flaccid as they call it. And the plant cell will wilt. So we want the plants to have water. And then the last one, guys, if the plant is in a hypertonic environment, that cell will get smaller and smaller and smaller, where the cell membrane will actually come in and pull away from the cell wall. The cell completely loses its shape, and we call this plasmolysis. All right? It's like the breaking down of the cell membrane. So not a good thing. What do I need you guys to know about this slide? Well, what is the best environment for a plant cell to be in? That's going to be a hypotonic solution. We want the plant cell to be in a hypotonic solution. That way the cell is nice, plump, or new vocab word that we learned, turgid. Okay. So that goes on with the bottom there, guys. You can see in the last picture there, when the plant cell is in a hypertonic environment, hypertonic solution, that different color in the back, okay, there, like the darker brown, that's the cell membrane actually pulling away from our cell wall. All right, so we'll do facilitated diffusion and then we will call it a day. So facilitated diffusion, it's pretty much like regular simple diffusion. Difference here is simple diffusion moved small, nonpolar molecules straight through those phospholipids of our cell membrane. Facilitated diffusion, on the other hand, these are going to move larger polar molecules. it's still going to go from an area of high concentration to an area of low concentration. So it's just like simple diffusion in that manner. Our difference is, instead of it moving through the phospholipids, it's going to move through some type of membrane protein. So here is our two different membrane proteins. We have our channel protein, okay, we talked about last time. They're able to go right through. Here is our carrier protein. Remember, they open up on one side, they close on that side, and then they open up on the other side, whether that's into or out of the cell, in order to let materials pass through. Okay. It's just like simple diffusion, still no energy is needed in order to accomplish this. Okay. So let me draw you a little picture of facilitated diffusion. Okay. And I'm going to do it with respect to our cell membrane and what it looks like here. Okay, so here are our phospholipids of our cell membrane. Let's draw some fatty acid tails in there. Okay. Now let's draw our protein. This is what it would look like from the outside. If we want to pretty much like do a cut in the middle of it to see what it would look like on the inside. Okay. Now, if you look at this, most of our membrane protein here, this area right here is going to be hydrophobic. How do we know that? Well, it's going to cling to our fatty acid tails here. If it was not hydrophobic on the outside of it, it would literally just slip right out of the cell membrane. Okay? So, let us draw some solute particles. Okay? We're going to say these are glucose. Okay? And we're going to say this is an environment inside the cell on this side, and that's an environment outside the cell. Now, glucose, large polar molecule, C6H12O6. What's going to happen is the glucose is unable to pass through our phospholipids like the oxygen was. So it was unable to do that. Instead, what's going to happen is they are going to go right through our channel protein to the other side. Okay. Now, how do they know this is the right channel protein for it? Well, if you remember, we have those carbohydrate tags, okay, type of glycoprotein, in order to make sure that, hey, is this one for glucose? And if it's not, then we got to find another channel protein somewhere else that is for glucose. So this is your difference between facilitated diffusion and simple diffusion. Again, simple diffusion, we're moving from high to low concentration. Facilitated diffusion, same thing, high to low concentration. Simple diffusion, we do not need energy. Same thing for facilitated diffusion, we do not need energy. The difference here is that simple diffusion moves our particles right through the phospholipids because they are small and nonpolar. Facilitated diffusion is going to move them through a protein because they are large or they are polar. All right, guys, so we will go over next time our active transport, which is our last type of transport, and we'll get through that. All right, guys, have a good rest of your day.